Okay, welcome everyone to this webinar, and thank you for joining today. Today's topic is data science, OMICSLOGIC data science. So how is data science different from bioinformatics? And what we typically speak about is bioinformatics and application to different types of data or to different domains, biomedical data. Today, we are going to be talking about the more technical aspects of this upcoming program and how you can benefit from actually gaining a lot of the technical skills in coding, in statistical analysis and machine learning, and really see the application of these methods, not just at the conceptual level, but actually from learning about why and how to pick specific methods all the way to implementation. So I will be introducing the program, but together with me, Dr. Harpreet Kaur, Clinton Kunha, and Dr. Mohit Mazumdar will be taking on different sessions. And so before we begin, I will just briefly introduce OMICSLogic and what is it that we typically do. So as I mentioned, our focus is on biology as a data science. So we speak a lot about the applications of data analysis to high throughput data and training that we provide to elevate the level of biomedical research and also enable research by providing specific benefits and application to oncology studies, infectious diseases, neuroscience, as well as agriculture. And so one of the connecting dots between all of these are the applications of data science methods. And so today we'll talk a little bit more in depth on that topic. But typically, we provide research enablement through analysis of big data. So we actually have big data analysis tools that are designed to address challenges in processing, analysis, visualization, and integration of different types of omics data and phenotypic data. We provide mentorship where you can interact with our mentor community and actually get one-on-one private mentorship if you need in application to different types of data. And we also provide asynchronous training resources. So we have a collection of courses and programs organized with collaborators and different types of university, high school level, as well as college level training that we provide. So let me go on and briefly introduce this topic of data science. So what exactly is data science? Data science is the ability to work with data, so manipulate data, organize data, and that is informatics. And then to apply meaningful statistical analysis techniques to this data, which include different types of machine learning and statistical analysis and even visualization. So it's really analysis of that data. And then finally, you have this application of these methods and of this data to a particular domain or a specialization. And when we speak about data science, in general, data science tends to be agnostic. So data science methods, just knowing how to run analysis, could be applied to very many different domains: clinical, business, Internet of Things, finance and marketing, biological research. But specifically, if we talk about biological data, there's going to be a specific additional level of specialization that we need to discuss. And that will be the way that data is generated, and therefore, what are the specific questions that are being asked of that particular data set, and how can we later on interpret it? Because as opposed to other types of data, a lot of times biological data is much more challenging to interpret and to understand the technical versus the biological patterns that we find in this data. And those are critical to distinguish as we seek to find interpretable, meaningful insights from this kind of data. So the OmicsLogic data science program is designed to provide an overview of statistical analysis and machine learning methods, to discuss experimental design and some of the technical variation that we need to address before the data is analyzed, how to mine data for meaningful patterns, and what do we mean by associations, and then also the application of complex biomedical or biological systems using big data. So how do we infer these relationships from data, and how do we interpret them in the context of this complexity of biological systems? And so specifically, this program is going to be focused on machine learning for biomedical discovery. Specifically, we'll talk about what data science and bioinformatics are designed to do and how can you learn the conceptual approaches, but also the practical skills. We will talk about data processing, cleaning, and exploration, the application of data science in biomedicine. So what types of projects and what types of examples are going to be meaningful for this kind of analysis. And then we'll introduce machine learning algorithms for next generation sequencing. We'll talk about data mining in the context of X-omics integration, or the integration between different types of omics data, and how to apply deep learning and clinical phenotype annotation for some of the advanced methods of analysis that are being discussed and sometimes used in the context of omics data. Okay, so I see that a couple of messages here that people are mentioning that they cannot hear. Unfortunately, I think that is probably because of your connection or settings. So if you could try and rejoin, and so Narika, maybe you could post this message also in chat if people cannot hear. So specifically, I think that in the context of biomedical discovery, right, we're talking about the different aspects of biomedical discovery where machine learning and the analytics have to be contextualized, because we are going to be trying to connect the research question or the clinical or biomedical need and challenge to different levels of statistical analysis and exploratory analysis that allow us to connect this to the type of data that is being generated that essentially represents some kind of a biological experiment, even though this experiment could also be a design of a data collection without a specific experimental condition, but more in the real world sense. And so specifically in the real world sense, what types of data are we typically talking about? So as we move into this era of precision medicine, we typically mean that there is some kind of a phenotype at the top of the iceberg, at the level where we can see and observe the symptoms. And then underneath, we have this molecular data that we need to connect to the clinical data in order for us to be able to integrate the signal and extract some meaningful results. And so obviously in the context of biomedical discovery, we're talking about biomedical applications that include treatment and selection of treatment options, stratifying risks for patients, but also we're talking about the application for drug discovery and for clinical trials. And so in drug discovery, this analysis of big data from clinical data as well as from high throughput experiments can speed up the development and the translation of discoveries to the use by patients. But also in clinical trials, it's an important component to be able to select the right kinds of patients, to define the right kind of strategy for outcomes, and also to translate other findings that are beyond the objectives of the specific clinical trials. In other words, even if clinical trials have failed, to a meaningful insight for the next round of clinical trials or for modification. And so a big trend in both of these is the discovery of biomarkers. And these, of course, are markers of specific conditions, markers of specific biological processes, and we are looking for more and more precise markers. Oftentimes, what that means is that we are not relying on a very clear-cut signal, but a complex signal. And that complex signal is defined by the complexity of omics data, which could be, of course, of many different types and has many different ranges of values that we can use to combine into a signal. And that signal would be a integrated signal, essentially, from multiple types of omics data. So what kinds of omics data are we going to talk about? We're going to talk about phenomics, which is the characterization and the features coming from image analysis, from clinical data, from cell morphology. We're going to be talking about genomic data, or the mutations or collections of mutations found in the DNA. We will be talking about epigenomic signal, where we'll discuss ChIP-seq data, whole genome bisulfite sequencing data, transcriptomic data, including gene expression, levels of gene expression, transcript expression, and alternative splicing. And we will talk about specific examples where we will use this data to actually discuss the objective and the type of data that could be analyzed, because every type of data comes with a specific set of parameters that we need to consider when we are trying to set up and plan our analysis. And of course, one of the major challenges in this whole field of omics is that the data is actually unstructured, right? So as you look at the availability of data and the type of data that is being generated, more and more, this is going to be unstructured data, because structured data requires processing. So if you think about collection of biological samples from patients, or even generating that data as a high throughput experiment, you end up with unstructured raw reads. You look at different types of environmental conditions. And because of that, the first step that we're going to be talking about is processing that data, or converting that data from raw to a structured format, from different types of biological material that can generate digital data to an actual organized table that we can work with. And so later on, I'll talk about some of the formats that that data is stored in, so that we can discuss some of the more technical structures for this data that we will be looking at. But ultimately, the objective of data science is to provide guidelines for different steps of data analysis, which includes data processing, as you can see right here, exploratory analysis, and the actual data analysis, or comparative data analysis, or sometimes modeling of that data. And so to go from experiments to results, those three steps is what we will have to cover in this program, and talk about data cleaning and processing, formatting it for the specific type of analysis that we would like to do, exploring data using visual methods and tools, as well as looking at different types of approaches for data mining, data analysis, where we consider different statistical, machine learning, and deep learning models, and then inference. How do we actually test our findings and go back to where we started with, so that we can contextualize the findings and actually interpret meaningful results. So what are some of the typical challenges in this process? Some of the typical challenges include analysis and visual exploration of data, where we can use things like principal component analysis or clustering to really identify some meaningful groups of objects based on all of the available data, or based on an extraction of data. Oftentimes, this also means looking at taxonomic relationships, where we can combine biological understanding of where the data came from with our ability to structure different types of groups of data in the patterns that we find. So a lot of the processing specific to biological data actually combines those two methods. One is just exploratory analysis on aggregate of all the data that we have, including noise and signal, filtering for specific signal that is statistically significant, or integration between taxonomy, which is a biological concept or a construct, and the actual analysis of similarity based on the data availability that we have. So because of that, we are really looking at this collection of different types of data that also includes other types of data, and that could include imaging, clinical data, chemical data, phenotypic data, and all of this data has to go through this process of preparation and organization of this data into structured formats, which typically in the context of data science is going to become some form of a data frame. And so the first thing that we will do in this program is we will discuss how to prepare different types of structured data objects, data frames, vectors, matrices, arrays, and lists from the data sources that we prepare in the processing pipelines. And then we will talk about how to meaningfully organize this information so that we can look at relationships between categorical data, continuous data, and other types of data that we need to organize in a visual format so that we can meaningfully spot some results that are of interest for our analysis. So sometimes this means just summarizing the data that we have. Sometimes it's looking at patterns overall. A lot of times it leads us to the need for data preparation. And data preparation means that oftentimes data comes in a format of different statistical properties that we have to change before we actually can run an analysis designed to deal with a certain data distribution. For example, oftentimes when we look at next generation sequencing data for transcriptomics, we need to first transform it into a log normal scale to have a more normal distribution. Of course, this is not a universally applicable method because when we come to data sets that are, for example, metagenomic data, there typically is not a need to make that transformation, but instead we need to choose a completely different method of analysis to extract meaningful patterns. Now, also when it comes to exploratory analysis, we also need to understand that the majority of the data that we have has many different components and many different sources of signal that we could consider, including technical, including statistically significant, and including some non-statistically significant, but nevertheless important for our ability to extract meaningful portions of data for downstream analysis. And so that's why we will also talk about different methods of statistical analysis, including t-tests, f-tests, ANOVA, regression, and other types of methods that will explain the relationship between variables in our data that we can ultimately use for analysis. And oftentimes we will find that the statistical analysis has limitations, and the limitations come from several different types of sources. So one is the variability in our data is complex, and we cannot capture that complexity using standard statistical techniques because of low p-values and kind of complexity of the differences between noise and signal. It also can come from just the types of data, because we might deal with many different types with many different variables that we cannot connect into a single data set. And so that is really where we will turn to the topic of data mining and classification, looking at many different types of methods that are typically used for analysis, for classification, and for feature selection. So we will look at different examples of methods like decision trees, support vector machines, hierarchical or k-means clustering, including clustering for big data sets like PAM, fuzzy, and dbScan. We'll look at linear discriminatory analysis and other types of methods that will help us use this predictive analysis for inference, for feature selection, and something that we can ultimately interpret. We will also dig deep into the algorithmic properties of some of these methods to really understand how different approaches can help extract features that are responsible for discrimination between different types and different classes. And so we'll talk about methods of feature selection, including support vector machines and random forest for feature significance, as well as greedy methods of analysis like stepwise analysis of linear discriminatory analysis and such. We will also talk about an obvious topic that many are interested in, which is deep learning applications in omics data. So we will talk a little bit about different approaches for deep learning, how we can discriminate between different types of network topologies, how we can optimize some of the parameters, and ultimately, what are the applications? Where do these methods work well, and where do these methods do not work well? And how do we evaluate the performance, and how can we select the right type of method for the right type of project idea that we have? Now, importantly, many of these methods come with ready-made packages, and with practical exercises that you will be able to run. And so we have prepared a collection of scripts and curated data sets where you will be able to try and run the specific analysis of your choice, understanding the methods and their application in the context of what's called a package that might combine many different specific scripts and algorithms to extract some meaningful insights that is considered to be a standard in a particular field. As you can see here, this is the application of non-metric dimensionality scaling in MDS for taxonomic analysis of microbiome data. So we'll discuss these methods that really tend to combine multiple methods of visualization, of processing, of data analysis, and then translating that into biologically interpretable results. Now, what we will do in terms of your technical skills is we will introduce a number of different techniques and technologies to allow you to understand and appreciate the implementation of these methods. And so for coding, we will use both R and Python. We will introduce you to a number of different environments where you can run this code, including integrated development environments like RStudio or Visual Studio for R and for Python, respectively. We will talk about sharing your code. How do you effectively share your code using Jupyter notebooks, how to post your data on GitHub, how to create Google Colab notebooks that you can share and run your code in those as well. But ultimately, you will get a sense or a landscape of these different methods and tools, including some commonly used libraries and packages like the base in R or ggplot, specific packages like ggfortify and ggpubr. You will be able to look to use Python pandas, NumPy, and sklearn packages, including visualization in matplotlib and Seaborn. And you will be able to apply general coding practices that are going to be important as you develop your skills in bioinformatics. And that includes organizing your scripts using comments and functions, setting up the development environment, dealing with errors and troubleshooting your code. So learning how to interpret the errors and finding the reasons for the errors, and then how to prepare your data summaries and submitting curated data and metadata tables to sharing repositories like on GitHub or Dataverse. And we'll use Jupyter notebooks on GitHub and Google Colab, including we'll create some interactive visualization using Plotly. So these are the topics that we plan to cover from the technical side. But to really draw the connection between the technical and the conceptual, we will utilize a number of projects. And these are curated projects that our team has gone through, has annotated for the specific methods that the authors of peer-reviewed articles have used, and extracted some examples from these data sets so that we can try and practice on them, both learning about the usefulness of a method, but also being able to understand why this method is being used and how to interpret the results or how to use the results from such a data science approach. Now, these different projects include many different categories, so many different data types, including genomics, epigenomics, metagenomics, transcriptomics, many different types of projects, like projects in neuroscience and cancer, infectious diseases. And ultimately, this also will be applicable to a project that you can independently develop, because once you understand the data and the needs to transform and understand the data for analysis, you can then actually apply the same techniques to a data set of your own. Now, that data set could be found on the public repository, or it could be something that you actually are already working on, so your own data set as well. Okay, so let me pause here and try to address some of the questions. So if you have any questions, please feel free to post your questions in the chat, and I will go one by one and address some of the questions. So the first question that I see here is about different programming that I have used or that we will use in this program. So first of all, I will go through several examples in today's session of processing big data using the T-Bioinfo platform, how to download from that portal some R scripts that you can modify and use. Then I will introduce you to a number of scripts in R and in Python so that you can see how you can learn those as well. And then the other question that I see here is, can we publish our results in journals using secondary data? So obviously, yes, you can. There are many examples of reanalysis. It's called meta-analysis or reanalysis of existing publicly available data sets. Typically, about 95% of the actual data is not being published. It's only the 5% of the most significant, exciting, or relevant findings that people tend to put in the journal article. But that means that 95% of that data is still available for reanalysis. And oftentimes you can actually take and combine multiple data sets, integrate them into a new data set, and ask a completely different question, or even perform the similar analysis using different methods. And that oftentimes will lead to novel insights, might mean to something, might mean that you can find something that could have some significance. And if you do, then you can actually share that in a journal article. Of course, we do not commit to helping you go through the submission. We just can help you by training and offering you guidance in how to develop a bioinformatics project. Okay, so Hadia is also asking if the course will be recorded. Yes, I will come to that in a second. This session is being recorded. It's also being live streamed on YouTube, so you can always find it on our YouTube channel. But I will talk a little bit about the sessions that we will cover in the full program. And so those, yes, will be recorded and shared with all of the participants. Are there basic courses for a biologist who doesn't have computational skills, such as R and Python? From Linga Raju. So yes, I think that we live in an era of data science. And so it's critical for anyone who has an interest in applying data analysis to large data sets to learn gradually some of the required data science skills, including coding. And so that's why this program, I think, will be also useful for those of you that do not have the skills to code already, but want to acquire those skills. So that's really what the program is about. I think it's also going to be useful for those of you that do know some coding, but would like to learn a little bit more about machine learning, deep learning, and how to visualize and explore your data. And so I think to explain how the program can achieve all of that in a short time, I would like to demonstrate to you how the program will run and what are the different components of knowing how to ask a question, being able to access big data sets and integrate them, and then how to apply some of these data science skills to also explore and use machine learning on those data, and then demonstrate your results, share your results with others. Okay, so if there are no other questions, thank you, Durba, also for your interest. I think there are many applications in agricultural sciences as well. We do have another session that's going to be coming up about agriculture and the application in agriculture. But today, let's continue and talk about this general field of data science and see how we will achieve all of this during this upcoming program. So the upcoming program will combine 3 different things that you will be able to do. And the first one is interactive sessions. Just like today, you will be able to join a session on Zoom, and one of our mentors, myself, Clinton, or Harpreet, will be guiding these different sessions that you can interact with us and with others, ask questions, get help with some technical challenges that are being, that you're getting stuck with. And then also to get the assignments and the review for your completed assignment. So that will be covered in the interactive sessions. Included will be some self-guided study materials. So these self-study materials that we offer is something that I will show in just a few minutes. And then we will also use some tools. And these tools include the T-Bioinfo platform. Many of you are already familiar with that. It's a logical and user-friendly way to analyze all kinds of omics data, including genomics, transcriptomics, epigenomics, and metagenomics. And then we will turn to our coding portal, where you will be able to try different scripts, including scripts that you've used on the platform. I'll demonstrate that in a few minutes, so that you can understand not only how this platform works, but also what is the specific code and how to modify that code and practice so that you can test whether you understood or not. So I will demonstrate that in a second. So